This video is going to cover Chapter 11, which describes the process of cell signaling. Now, cell signaling is basically the processes that cells use to talk to one another in order to accomplish common goals. So cells have to decide what signals to produce, what signals they want to listen to, and then how to respond to those signals or those cues. This is really important for lots of major physiological processes that includes development, how we go from being an embryo to becoming an organism, apoptosis or programmed cell death, which we've talked about a little bit in class, as well as immune system recognition and how we mount an immune response. Cell signaling is really, really important in lots of different processes, and also when cell signaling becomes dysregulated, that can lead to many different diseases, including cancer. So you should have already watched the pre-class video, which covers these 4 types of cell signaling as well as synaptic cell signaling. So make sure that you can recognize different types of cell signaling given a specific example. What we're going to cover today first is the process of cell signaling. So it can be broken down into 3 major phases. First is reception or receiving the signal. We're going to talk about 2 kinds of receptors. Intracellular receptors we'll mention briefly, but then most of our focus is going to be on cell surface receptors. Then we're going to go through transduction, which is transmitting the signal through the cytoplasm as well as amplifying the signal, and then finally the specific types of responses. But the major focus of this chapter will be looking at the pathways that are regulated by 3 major types of receptors: the receptor tyrosine kinases, the G protein coupled receptors, and the ion channel linked receptors. So we'll go into these signaling pathways in great detail, and I will expect you to know these pathways. You should also complete the worksheet or activity that I've included with this video lecture. And then finally, in class on Monday, we'll look at how these pathways integrate in apoptosis. All right, so let's start by talking about reception. So as we mentioned, reception is essentially recognition of the signal. So here the signal is referred to as the ligand, so that's just another name for the signal molecule. So this signal molecule can be referred to as a ligand. That's Ligand. And there are 2 types of signals that a receptor can recognize. So first we can have a hydrophobic signal molecule. So hydrophobic signal molecules, these could be things like steroid hormones that can easily pass directly through the plasma membrane by diffusion. So these diffuse from the environment through the plasma membrane, and then they are recognized by an intracellular receptor within the cell. So the intracellular receptor is located in the cytoplasm, and when it binds its ligand, then it becomes activated. And these steroid hormone receptors can then enter the nucleus where they activate gene transcription. So this is really important for hormones like testosterone or estrogen, that when they activate their receptor in the cytoplasm, so here we see the cytoplasmic receptor, the receptor then activates a bunch of different genes. Essentially, it acts as a transcription factor to promote transcription of genes relevant to that hormone. So, for example, for testosterone, it may promote muscle growth. So that's all we're going to talk about: intracellular receptors and steroid hormone receptors. The rest of this chapter will be focused on our cell surface receptors. So cell surface receptors, instead of binding hydrophobic ligands, they bind hydrophilic ligands. Because these ligands are hydrophilic, they cannot cross the plasma membrane unaided, meaning that they have to be recognized at the surface of the cell. So these are hydrophilic signal molecules are hydrophilic ligands, and they bind these receptors called cell surface receptors that are embedded within the plasma membrane. Then this cell surface receptor has to send the signal to the inside of the cell, and it's going to lead to some kind of response. So let's look a little more at the structure of these cell surface receptors. We can break them into three parts. So first we have the extracellular ligand binding domain. So this is the part of the receptor protein that is outside of the cell, so it's facing outward, and it can bind the signal outside the cell. So it binds this ligand, and the ligand has to be complementary to the receptor. So they generally match in shape, and they're electronically complementary as well. So for example, if the ligand is positive, then the receptor would have a negative charge in the binding site. So the next part of the cell surface receptor is the transmembrane domain. The transmembrane domain is a hydrophobic part of the receptor that is embedded within the plasma membrane, and it transmits the signal to the cytoplasmic domain, and those cytoplasmic domains can vary, but they always have some portion that becomes activated in order to transmit that signal into the cytoplasm. So when the receptor becomes activated, it's going to change something in this cytoplasmic domain in order to trigger a cellular response. Many times this cytoplasmic domain acts as an enzyme, so that when the ligand binds, it changes the shape of the cytoplasmic domain, activating the enzyme that then in some way transmits this signal into the cytoplasm. So now let's talk about this second piece. So the first piece we've already talked about, reception. So if we have a cell surface receptor, the receptor receives the signal, but the second phase was transduction. In the transduction phase, what we're doing is passing the signal from one molecule to another, almost like a relay race. So the receptor becomes active, it activates another molecule, which activates another molecule, which activates another molecule. Then the other part of transduction is that the signal has to be amplified. So what I mean by amplified is that you're getting more and more molecules involved in the signal. So maybe the receptor activates 5 molecules of this molecule A. And then molecule A, each one activates 5 molecules. So now we have 25 molecules of molecule B active. And then if each one of those activates 5 molecules of molecule C, now we have 125 molecules of molecule C that have become active, all due to the action of one single ligand binding a receptor. So amplification is getting more and more molecules engaged in this pathway in the cytoplasm. What I want to talk about next are what are some ways that we can amplify the signal. And there are 3 major ways that we're going to see that we can amplify the signal. So the first way we can amplify the signal is through a kinase cascade. And hopefully some of you will remember that a kinase is an enzyme that adds a phosphate functional group to another molecule. And a kinase cascade is activation of a series of kinases through phosphorylation. First, imagine that we have a kinase here, so this kinase protein becomes active due to some upstream activation. When it becomes active, it phosphorylates, adds a phosphate group to the second kinase. That then activates the second kinase, which phosphorylates the third kinase, making it active. So now your third kinase is active, and your third kinase can go on and phosphorylate something else to have an effect. So in this kinase cascade, you can imagine that the first kinase would activate many molecules of the second kinase, which would activate many molecules of the third kinase. So we're getting more and more molecules involved in the signaling pathway at every single step. Another way to amplify the signal is through the use of a second messenger. Another name for the ligand or the original signaling molecule is the first messenger. So a second messenger is a molecule that's produced by an enzyme inside the cell that can also be used for amplification. And that's because an enzyme can make many, many molecules of the second messenger. And so there are several examples of second messengers that we're going to see. So the first example is cyclic AMP. So cyclic AMP you probably already recognize from your lac operon knowledge. So cyclic AMP was the molecule that activated CAP in the lac operon, and in that case cyclic AMP was actually acting as a second messenger. So cyclic AMP is created by an enzyme called adenylyl cyclase. So adenylyl cyclase is the enzyme, and its substrate is ATP, and it takes that ATP and removes 2-phosphate, so it removes pyrophosphate. Adenylyl cyclase then creates a bond between this phosphate and the hydroxyl group at the 3' position. So it creates this bond. So adenylyl cyclase is the enzyme, and it produces cyclic AMP from its substrate ATP. So another example of a second messenger, there are two examples here. So IP3 is inositol triphosphate, and DAG, diacylglycerol. So the enzyme that produces these molecules is called phospholipase C. And what phospholipase C is, it takes a lipid substrate, PIP2. PIP2 is phosphatidylinositol phosphate, and it cleaves it. So you can see right here we have this is a phospholipid with a large head group. So what phospholipase C does is it clips the bond between the phospholipids, so that leaves diacylglycerol still embedded in the membrane. And then IP3 is the head group. So IP3 is then released into the cytoplasm. And both of these can act as second messengers. So once again, phospholipase C is the enzyme, and it produces the second messengers, IP3 and DAG, from the lipid PIP2. And a third second messenger is calcium. So calcium is stored in the endoplasmic reticulum, and calcium isn't exactly produced by an enzyme, but it can be released. So calcium channels in the ER can release calcium into the cytoplasm, where it can act as a second messenger. So our 4 examples of second messengers were cyclic AMP, which was produced by the enzyme adenylyl cyclase, IP3 and DAG, which were produced by the enzyme phospholipase C, and calcium, which was released by a calcium channel. The last method of amplification, so we've talked about kinase cascades and second messengers. The last mechanism of amplification that we see in cells is a proteolytic cascade. This is actually a lot like a kinase cascade, except how the molecules become active is by cleavage. So a good example of this is a caspase. Caspases are enzymes that become activated when they're cleaved. It's almost like removing a protective cap. And when they become active, they cleave other caspases and activate them. So that's the cascade. So this is an example of an inactive procaspase before it's been cleaved. And when it's cleaved, it removes this inhibitory domain that makes it active. So now we have the activated caspase. When that caspase becomes active, it activates the next caspase by cleaving it, which activates the next caspase by cleaving it. So we get more and more caspases that are active, so we have amplified the signal. So finally, I want to talk generally about the different types of signaling responses. But the first thing to note is that responses are specific to different cell types, and that the same signal may have different transduction pathways in different cells. So one signaling molecule may activate a linear path in one type of cell, may activate a branched path in another type of cell, and sometimes different pathways can converge upon one another. So that's what leads to the broad variety of responses that we see in different cell types. But in general, there are three major types of responses that we see. One type of response that we see is a change in metabolism. So that could be breakdown of the carbohydrate glycogen, which is stored in the liver, to glucose to increase the levels of glucose in the bloodstream. We also can see changes in gene expression. So that would be activation of gene expression or repression of gene expression. So everything that you've already learned about how gene expression can be turned on and off can happen in response to a signal. And we can see changes in cell motility. So that may involve rearrangement of the cytoskeleton. This is really important for processes like angiogenesis, which is the formation of new blood vessels. So the cells that are going to form the blood vessel need to move in a specific direction in order to form the new vessel. So just to review, the three different responses were changes in metabolism, changes in gene expression, and changes in cell motility. So I'm going to end this video here, and then I'm going to start a second video to start going through the three major signal transduction pathways.